So good morning, everyone. So welcome to the second session of the What's Up with MALDI-Tof MS Spectrometry in Microbiology. Welcome to the new participant. Welcome back to all the people who were here yesterday. So for people who were not here yesterday, I'm Maureen Fuscherl, and I will just host this session today. So like yesterday, we're still pretty international with a lot of people. Before to start, I would like to thank all the speakers to be here. Really great person. Speakers, which is in fact the keynote speakers, it's Professor Ray Kruse Iles. So he has a PhD in molecular pathology from the Barts in London Hospital Medical School. Currently, he's a clinical scientist, former university dean, and author of more than 150 peer-reviewed publications spanning reproduction to cancer. Through recent advances in mass spectrometry, his team has developed techniques and expertise in proteomics and basis of biomedical startup companies pioneering post-genomic, faster, efficient, and cheaper diagnostic screening tests. So, Ray, please, the floor is yours for your presentation.
Maureen, thank you very much for the kind introduction, and I'm smiling because this symposium's been fantastic. I feel I'm with my people at last. Every speaker has said something extremely relevant to what I am doing, and I feel really amongst the people who understand the kind of technology that my colleagues and I are trying to promote, and obviously you're doing exactly the same thing. If I come into my talk, I'm really going to talk about the work we've been doing over the last 12 months on COVID, the identification of SARS-CoV-2, but it is an example of how the mass spectrometer can be rapidly applied and I think will be the biosecurity screen of the future for future pandemics. I realized when I was putting this talk together that I put why and how the wrong way around, and I'm going to start my talk with the why. Why is MALDI-Tof going to be important? Hopefully this is going to go. So if I click first. Oh. So why do we need a MALDI-Tof spectrometry test for SARS-CoV-2 and other viruses? Well, at the moment, or what was very fortunate, we had RT-PCR viral genome testing, and that really stepped in to give a handle on this pandemic and essentially put everyone in the right direction to get this under control and for us to actually monitor this outbreak before the vaccines have started to come in. So it was our main way of knowing who was infected. The problem is it's time consuming, we need specialist labs, costly, but it is highly sensitive. And this is an issue that I'm going to elaborate on in the next few slides. Now, what has been introduced is this lovely little bit of plastic called a lateral flow antigen test. The use of the lateral flow antigen test in the UK has skyrocketed, a little bit to the detriment of PCR. And it's rapid, simple, cheap, but the trouble is it's got a low sensitivity. Now there is a bit of a joke amongst lawyers that there's nothing as expensive as a cheap lawyer. I hope for people's lives that there's nothing as expensive as a cheap diagnostic test, because what I'm going to illustrate to you is that what is needed is a highly sensitive test which can pick up people who are not presenting with symptoms. In particular, we want to pick up the asymptomatic. Asymptomatic are people who might have the infection, they're able to deal with it, but they pass it on to other people, some of who will develop the symptoms and become ill. So you need a highly sensitive test, but at an affordable cost that you can screen entire populations. Now, another bit of an argument is, to be honest, I think to be reliable, it needs to be conducted in a laboratory, not by individuals. But this is my personal view. But there is a real problem with this real world pandemic. Detecting the asymptomatics is very difficult. It is not something that can easily done by a bit of plastic. And we also have imperfect data sets, as Kathleen was talking about, in that we use RT-PCR to define who is positive. But it's not the perfect definition of who is infected or clinically infected with COVID or SARS-CoV-2 that they are going to spread it. In fact, there have been very many studies, and they're saying that RT-PCR is great. It is a very, very good tool, but it's only about 85 to 90% sensitive in picking up all the people who subsequently develop symptoms from COVID-19. The issue of false positive had been largely ignored, but it's really come up. The reason is that we're all now starting to screen people who haven't got any symptoms, they're not presenting with symptoms, so we've got to look to try and identify the pre-symptomatic and then that true asymptomatic population. And the estimates of this pre-symptomatic asymptomatic population can range from 4 to 80% that are being identified by RT-PCR. But most studies will agree that RT-PCR, as it's applied to the general public, not necessarily presenting with any kind of symptoms, that about 50% will be asymptomatic. They will not develop the disease, and we're presuming that they are able to spread it to others. So nevertheless, you know, we really need to do this. We need to identify the asymptomatics, and the lateral flow device has been sort of rolled out in the UK in particular, and it's about to be rolled out in the US as sort of the rapid screening technology to screen the entire population of people who do not have symptoms. Well, the problem is that this test was only developed and validated against people with symptoms in hospital. And when it started to be tested in real life, and in particular the Birmingham study, where they were looking at students, over 7,000 students who did not have symptoms, they only picked up about 3 to 4% that were being picked up or should have been picked up by an RT-PCR positive test of the non-symptomatic population. So that is extremely disturbing. And in fact, the data really, really points this out. You're able to download the statistics from the UK government website, look at how the what's called tier 2 screening is working, where the PCR test is looking for primer pairs, looking at the M protein, ORF1AB, and then the S protein. So we're going for 3 target gene sites of the virus, and you only need one of them to score positive within the PCR for you to be labeled as being infected with SARS-CoV-2. And there's also a big question about the kind of level that is being detected. And what has been shown on many, many studies is that the lateral flow device will only detect high levels of virus if it's present at CT values less than 29. To be honest, a value of about 26 is probably about the average for the lateral flow devices. So as you can see from this graph here, this is the UK government's own data from their surveying of random surveying of the population, that you're only picking up about 25%, maybe 30% of the people that are detected by the RT-PCR, and presumably these people here are more likely to be the asymptomatic population, and you're completely missing them using the lateral flow device. One thing to remember, that we are talking about huge log changes here, that every 3.3 additional cycles, you're moving a log difference in sensitivity. So if you're detecting at about a level of, say, 100 units, you're measuring in tens of units if you jump 3.3, And you can see how this rapidly gets your sensitivity down to very, very low levels. And there is an argument with the RT-PCR that if you go on to too many samples, you're measuring noise, possibly environmental, of no clinical significance. So some PCRs limit their CT value runs to 35, as we do in Europe and the UK. In America, it's 40 cycles. Now, the data really has shown that the lateral flow tests, when we looked at the cumulative data of all its uses of it, it's been used, over 7 million tests have been reported, that it was reporting about a prevalence of about 0.1% positivity. Whereas if you look at the equivalent RT-PCR data, it ranges from 0.3 to 0.6, and if I do the UK average, that's about 0.3. So it's not that the lateral flow tests for detecting a third of all positives. It's what third is it detecting, and the third it's detecting is the pre-symptomatic, the symptomatic fraction, and not the dangerous below the watermark asymptomatic infections, which would sink the Titanic. It is not an appropriate test, and that's my argument. So, MALDI-Tof, how can this come in and help be an adjuvant, high throughput screening test that can support RT-PCR? Well, the first thing we've got to do is actually think about what our target is, and it's really about the physical biochemistry, the biology of the molecule, and something that Kathleen was saying, that you really have to look at the biomarker you're going to be looking for. Now, unlike bacteria, there are no housekeeping genes. There are no naked small proteins which will be characteristic that we've seen in a lot of the previous talks. The virion particle itself, the really characteristic proteins are glycoproteins, which are very large and embedded into the lipid envelope of the virion particle. And these are the key activators, the key players in the infection of a cell. They'll bind to a receptor, they'll be acted on by other factors such as a proteolytic trigger, which will activate the complex of the spike so that the viral nucleic material, the RNA, can be introduced and it can take over the cell. And as you can see here, this is an infected cell with a coronavirus, and you can see that it's completely taken over the cell function, and what it's doing is making its own protein, i.e. the viral envelope proteins here, and there's quite a few of them, creating them embedded within the lipid, and then it takes copies of the RNA and produces little tiny vesicles like endosomes, which are packed to the viral particle, and then the viral particle travels to the cell surface, and we get millions of these viron particles released. Now, we decided that MALDI-Tof can raise this challenge, and something Alex was saying is that, you know, we looked at the real life here. We couldn't grow our virus on agar plates and then pick a colony. We have to take them from clinical samples. So we have to consider several steps of developing a MALDI-Tof technique. So sample enrichment, and so we were looking at either swabs, or what we opted for initially is actually a gargle to try and get the mucus from the back of the throat where the virus should be in the initial stages of infection. Can we take a clinical sample, enrich the viral component, and then we had to consider, well, we've got a problem in that the proteins we're interested in are embedded within the viral plasma membrane, the lipid membrane. Can we get those out in such a manner that they can be then visualized on the mass spectrometer? Because if they're stuck within the membrane, we're never going to see them. So this is our first two challenges. And what we did was some very simple physical biochemistry. One of the things is just exploiting the physical size of the virion particle. If we add acetone, and we just add a 1-to-1 ice-cold acetone to our biological sample, we can precipitate the larger complexes, the larger molecules. It turns out if you do a 50-50 mix, you are precipitating and enriching into that pellet your virion particles, and you're leaving behind an awful lot of other smaller molecules which would give you interfering signals in your mass spectra. So we cleared an awful lot of smaller molecules. So once we'd got our enrichment of our viral particle, we then had to consider, well, how do we get out the characteristic proteins that we want to see in the mass spectrometer and get them into a mass range that can be visualized on the mass spectrometer. Now, we knew that we didn't want to over-process, and we also realized that from our own experience, we were looking at much larger molecules than everybody else has been looking at in this conference. We also had to change the matrix to look at these bigger molecules, these glycoproteins. We can't use CHCA, we have to use cinnapic acid. But we still had to try and break up and extract these proteins. So we actually had developed a series of mass spec, multi-mass spec friendly detergents, which will dissolve away the lipid membrane and aid in the ionization of these lipid loving domains so we could actually visualize the proteins coming out. And to make it a little bit easier for us, we also reduced the disulfide bonds. There's an awful lot of disulfide bonds here, which holding the complexes together and holding some of the structures together. And in particular, this spike complex, not only is it a trimeric complex that we could break apart with our detergents, our mass spec friendly detergents, but if we actually reduced it with something like dithiothriotol, which we have been using up until now, we could break the spike protein into several subunits. Typically people understand S1 and S2. What I'm going to show you is that actually there's many more tryptic fragmentation of the S2B, and that's a very important biologically functional protein. Now, I'm just going to come back in that all I've shown you to date or so far is very relevant for looking at other viruses. Many envelope viruses, in fact, all of our envelope viruses have some kind of spike or receptor binding complex embedded within the membrane. They're of different lengths and they have different endogenous and then subsequent cleavage points. So they all give a different fragmentation pattern. So this really gives hope that we can actually look at many, many different viruses using this technique. But I am going to concentrate on SARS-CoV-2 for the purpose of this talk. So to get all this developed, we couldn't initially start all the research using live virus. That would be dangerous. There's so many conditions we had to adapt. So luckily, colleagues at Kent University and University of Cambridge, absolutely brilliant people, were making pseudoviruses where they were had taken the components for a lentivirus. They could introduce the spike complex into the pseudo lentivirus, and that would be expressed in culture, and we would get produced into the culture media these pseudoviruses expressing the targeted spike complex. So we would treat it as if it's a biological sample. Our aim was to do a gargle sample, and we could actually then look at the spectra, and we were getting fantastic results from these pseudoviruses. Albumin could get in the way of culture, which of course is not present in a gargle. You don't have albumin floating around in your saliva. But we definitely were picking up the S1 fragment, which we confirmed. We're also picking up the S2 fragment, but sometimes that could be obscured by albumin. And then we were getting a host of the other large envelope proteins like E and M, the matrix ones, were also coming up as a signature. We then moved on to live virus. So we were getting the Wuhan isolate and the isolate from the UK. These were being grown on cells, and we started to play with that. We would take the culture media, not the cells, just the culture media where the viral particles were being shed into. We could spike it into gargle samples, and yes, we could see the S1 protein in particular. Now, I'm just going to point out a little thing here is that we're getting very broad peaks. The reason we are because these are very large proteins which are glycosylated. So not only do we get a broadening of the peak because of isotopic variation, because we've got carbon, nitrogen, oxygen, and sulfur isotypes. So that really causes a big broadening of your peak as you get bigger and bigger and incorporate more of these different elements with their isotopes. But also we get variable glycosylation of these peaks as well. So it tends to be very broad peak, and we could actually see a difference in the glycosylation pattern from cells which were grown on Vero cells. So when the SARS-CoV-2 was being propagated on SARS-CoV-2, when we started to move into patient samples, we could see that there was a glycosylation difference, and this peak was slightly higher. So it is a very broad and complex peak structures that we are getting on our mass spectrometer. So we have the basis of a test. Sampling could be at home. It's just a simple gargle, 10 mils of water, turned out to be better than saline. It could go to the laboratory where we would filter it to get rid of any bacteria, and then we'd do the acetone precipitation, put it onto the mass spec, we'd get a spectra, and we could then start to identify the various peaks. And one of the most interesting things was happened when we moved into real biological samples is that noticed that we were getting some extra peaks which were actually arising from the precipitation of the large immune complex molecules that you will find in your mucus, so IgA. And these were characterized as, because we were reducing the disulfide bonds, we could see the IgA light chains from the IgA heavy chains separating as well. And this turned out to be an additional important marker, because if you had viral proteins, you would have high IgA levels as well. So the elevation in IgA also told us that you had an active infection. It wasn't just some kind of noise or something that was not active, not doing anything to the body, that we can actually see an immunological response in that mucus saliva sample as well. And when we came to analyzing the spectra, we could see viral envelope proteins that could arise from a number of different viruses, because you've got a lot of bacteria in there, you've got lots of different viruses that may be present. You could see whether there's immune response from the IgA heavy chain and the IgA light chain, and then we were looking for the specific S1, S2, and S2-like fragments, which I'm going to elaborate on in a minute, present in your spectra. And we could do a heat map. We were looking at the bioinformatics that would be needed. We did some limits of detection to make sure that we were in the ballpark range of RT-PCR, and using the 8020 Shimadzu instrument, we were getting down to the upper ranges of some of the EUA, the emergency use authorization PCR tests in terms of their limits of detection. So we were in the range of what PCR tests were actually detecting. So we had a good start. But as Alex had pointed out, there's a big jump between what we can show in the laboratory and what we can do in real clinical life. There's a lot of development that had to be done. What we concentrated on optical is the optimization of the different mass spectrometers and the data processing. There is a lot more work that can be done on matrix formulation. We have just stuck with synapinic acid, but I think that there are many other matrices that we should be trying to see if we can get better quality spectra, but that's another project that we haven't even started. what we wanted to do was to see if the basic technology that I've described could work on any mass spectrometer. So clinically out there, we have the bioMérieux essential, the Axima. We have the new 8020 that was being produced by Shimadzu, and of course, there are many people out there using the Bruker Biotyper, the Microflex. So these are the instruments that are already out across the world, and we wanted to see if the technique could be worked or could be used on all these different platforms, because there are differences, as pointed out. Every machine has its own peculiarities. Now, the first one that I'm really pleased to report, this is fantastic work with our collaborators in Northern Illinois, Professor Elizabeth Galliard with her students Praj and Zane. We sent over our protocol, all our reagents, what to do. They sent Spectra back to us, and they were fantastic in that they had a program where all the student athletes who were asymptomatic, or should I say didn't have symptoms, were being tested by nasopharyngeal swabs and being scored whether they were RT-PCR positive or not. Surprisingly, 77 were RT-PCR positive, and 10 of which had developed symptoms. So there was a large percentage of this asymptomatic population, which is the key population to look at in this cohort that Beth and her group were studying. So the overall amongst that population, 12% were asymptomatic. There's 14% who came back PCR positive. Fortunately, amongst those, 152 gargle samples were collected simultaneously with the nasal pharyngeal swabs. And Beth performed the test that we were developing, first version of it, on the Axima instrument, and very grossly sent also the spectra back to us to sort of play with the mathematics of what peaks are important and which aren't. Of those 152, 3 were pre-symptomatic, so that was our sort of absolute control group in that we had to detect those at least. And then there was 57 RT-PCR positives, but they remained asymptomatic. They didn't develop any kind of symptoms. And then there were 92 RT-PCR negatives. Now, this is not a perfect yes-no situation. You know, please don't say RT-PCR is a perfect gold standard, but it is the best we have, the very best we have. We started to try and tease out the true positive and infectious asymptomatics from amongst that cohort by looking at the spectra. So in red here, we have the people who did develop symptoms, and the blue here are the RT-PCR positives, and the green are the RT-PCR negatives. Now, depending where you draw your cutoff, it could be here or here. And we decided amongst all of this analysis is that we wanted to see that you had an immunological response, an elevated level of IgA heavy chain, and whether we could pick up either the S1 or the S2 and its S2 derivative peaks, which I'm going to show you in a minute. And overall, we had about a 70% agreement with PCR tests, which is so much better than the agreement that was occurring with lateral flow devices. And this is significant. So we can pick up all of the pre-symptomatics in agreement with the Abbott SARS-CoV RT-PCR test. Depending where our cutoff and where you want to set this, because I think we're picking up a lot of noise. I think, you know, we might breathe in, doesn't take, you breathe out. There's a lot of environmental noise with a viral infection that's going around in the air. We can get varying amounts of positive agreement or negative agreement, depending on what your cutoff is. So back in the UK, without any UK government support, we looked at the data set that we were getting from Beth, looked at what samples we were getting in the UK, and we started to improve the system. The first thing is the data analysis and use of controls. One of the things that we really came very clear is that we need to have positive control to make sure that the mass spec system is picking up these high molecular weight molecules. We've got that. We then had to have a reliable marker of whether the sample given was sufficiently good. And we actually had that similar to something that Alex was showing, is that if we look slightly lower in the spectra, around about 10 to 11,000 Daltons, you pick up proteins which are associated with mucus. These are the non-mucus mucus-associated proteins. They are heavily cross-linked with disulfide bonds, and they hold the mucus together. Of course, because we were reducing everything, if we did not see these mucus-associated proteins, then we knew that we were not getting a very good sample. But it also enabled us to do some other things as well, is that, well, that gave us a little bit of a handle on quantification. So we developed an algorithm where we would sum the S1, S2, and S2 prime markers, which are markers of the coronavirus. We would normalize that against the peak intensities of these mucus proteins. So we've got a normalization going on. And it was sort of something equivalent to the PCR test, where they were looking at 3 different primer sets. Well, we were looking at 3 different markers. Yes, only one needed to be positive, but it had to be above a threshold. And our controls really started to get us a handle on how good the sample was, if there was any detector drift, because you do get detector drift as you're going across many, many samples. And we also had the additional control of, well, is there a oral mucosal immunological response going on as well as detecting these markers of the virus? So it's quite, at the moment, I'm quite pleased with myself, but or ourselves, but we'll see how things develop. Back in the UK, we did have access to a number of samples where we were getting gargles from. One of the projects I'm involved in is looking at the immune response using mass spec, multi-mass spec, for people who were serologically confirmed and PCR confirmed to have had SARS-CoV-2 infections. A large number of these were seriously ill on the intensive therapy unit, and the rest were medical staff, had mild symptoms. And we were looking at their convalescent samples. We got them gratefully to give us a gargle as well. And we could look and show that, well, actually they're clustered down in this bottom corner here, where it came to looking for the coronavirus peaks and the IG heavy chains. That could be variable, it could be other infections going on, but basically we could set some thresholds. We also looked at 300 members of the general population, volunteers between London and Bedford, just about the time when cases were surging within the UK. And we looked to see what kind of signal intensities we were getting from that population. And that was quite interesting because the majority were down within the low cutoff threshold, but we could actually detect a number of people, and the number of people we were detecting were consistent with the published RT-PCR data for the region, in that we should have detected something of 8 to 11 in our random sample. We were indeed, depending on the cutoff, we were picking up between sort of 70% and 80%, depending where you drew that cutoff values, which would match what would be the expected population of sort of pre-symptomatic, asymptomatic carriers within the population at that time. One thing to note is that unlike the American series, that the CT cutoff on the RT-PCR in the UK, as I think is the same in the rest of Europe, is 35, not 40. So it's got a lower threshold of detection. It's not got it's got a more conservative threshold for detecting a COVID-19. We also looked at how this would perform on the Bruker Biotyper, which is very widely used across the world. And this is great collaborative work from Erica and Julie in the UK. They helped us with how we could get the data off and analyze it as text files. We set up a different raster pattern because we don't sample a small number of spectra. We actually have many thousands of spectra accumulated per sample, and we have a specific well-defined raster pattern as we go across the spot, so we're able to put that onto the machine. We looked at optimized laser, detector, pulse extraction settings, and they're all set now for the Biotyper. And we also looked at the lens cleaning and the infrared source cleaning system on the Bruker is very good at this, and we adjusted sample shots so that we could maintain high efficiency, that we weren't getting over splattering of sample matrix onto the primary lens, which can then deteriorate the quality of your spectra. And this is the performance of the Bruker Biotyper. We also, I should have added that we changed our buffer formulation slightly because we realized that DTT, although it was having a fantastic reducing activity, the activity of dithiothiazole drops off with time. So if you're doing hundreds of samples and your dithiothiazole is out for a couple of hours, you'll find that its efficiency of reducing all of those disulfide bonds drops with time. And we moved over to TSEP, and we were getting much higher reproducibility. This very tight distribution. Again, we were looking at seropositive convalescent patients, this time at 6 months. Obviously, this is after we had done the work on the 80-20. We've only have a few 88, in fact, samples from general population at a time when the incidence of SARS-CoV-2 detection by RT-PCR has dropped dramatically within the UK. But we've got this nice clustering here, and the one positive is here. Unfortunately, you know, we haven't got control. We haven't got that support to confirm which is RT-PCR or positive or not, as we have in the US. Now, we seem to have a system, and that system looks like we could have some relatively straightforward interpretation software. The bioinformatics seems to work. We've got the controls, but the emergence of variants are the critical emergency that's occurring now in the world. And how would the MALDI-Tof analysis keep up with this changing field, this rapidly changing field in this pandemic? The variants all are interesting in that the variants are occurring within the spike protein, and as you can see that we target the spike protein subunits seen on the mass spec. They are interesting in that they seem to be clustering with particular mutations, and these are associated with increased spread and also with decreased antibody responses, that they are having effect on the structure of that spike complex, such that in some cases not recognized by some of the antibodies. Now, we have recently just entered an agreement with some fantastic team in Brazil, and we're just about to start our study using the developed MALDI-Tof mass spectrometry test for this viral infection in Brazil. And we're particularly, with this group, we are going to be looking to see how different the spectra is with these variants of concern, in particular the P1 variant. Now, I'm coming back to biology and the structural biology, because this is very important when we're starting to looking for these protein markers, you know, at limited levels, at very small levels within biological samples, and how they might be changing. This is the trimeric spike complex, and we already know that there is a pre-priming furin cleavage site here between the S1 and the S2 subunits. When binding occurs to the ACE2 receptor, we see that the entire S1 complexes drops off. So we have a trigger event exposing the S2 complex. Now the S2 complex suddenly changes its conformational shape in a dramatic sense. It's almost like an umbrella turning inside out. And suddenly it changes its shape and exposes fusion peptide, which binds into the membrane of the target cell. So here is the membrane of the virus, here is the membrane of the target cell, and the fusion peptide region has suddenly been exposed. It would be internalized normally, but a whole conformational shape has occurred. We think there's another cleavage site here, and very early on, using MALDI mass spec, we had surmised that there wasn't just an S1, S2 prime cleavage, this S2 cleavage that other people had recognized, that possibly there's another cleavage here that is responsible for these next stages of biological activity of this protein. Because once it's bound into the S2 complex is bound via the fusion peptide into the membrane of the target cell, you see there's another big conformational change where the upside down umbrella seems to pull backwards and folds back in on itself, so as to bring its own membrane bound domain and the fusion peptide membrane bound domain together, so that the nucleic acid material of the virus can enter into the cell. Huge conformational changes, and I think there's a lot of cleavage events going on there. This all dramatically changes how antibodies are binding and what is being recognized. Now, during the second wave that occurred in the UK, we were pushing our mass spectrometers to the limit to see if we could pick up asymptomatics. This is a 60-year-old asymptomatic. We could pick up our S1 domain, our IgA. We could normally pick up the S2, which is about 71, and sometimes we'd pick up another one about 62, which we think is where there's this additional cleavage going on, and there's a much smaller fragment being released. We started to see odd things, such as this one, where there's our S1, our broad S1 peak, there's all the other viral envelope proteins, but that S2 has shifted in position. It's now at 66, no, 71. Now, we didn't know what was going on here. We need more samples. And then suddenly we got this one, where this is a young female in her mid-20s. She was PCR negative twice, and then finally a PCR positive for SARS-CoV-2, and was hospitalized and treated with steroids successfully. But as we can see, we couldn't detect any S1 or S2 fragments in the positions we would expect, but we're suddenly getting all these new fragments coming up in samples. Now, because we haven't had quite the coordination I would have liked in the UK, I haven't got RT-PCR data on this, but this could be a completely different virus, or it could be a very interesting virulent new strain of SARS-CoV-2 that has arisen. But this just illustrates that we see the pattern changes on the mass spectral spectra when we start to run a gargle sample and look at it on the mass spec. There is very clear indication of a viral infection. I'm not sure what this is, whether this is SARS-CoV-2, which is a mutant, and we've got a different pattern now, or it's a different virus altogether. So the new studies that we are doing, we are hopefully soon going to be starting with Professor Dr. Tim Garrett at the University of Florida, looking at clinical samples, which will compare the Shimadzu 8020 with the Bruker Biotyper, looking at saliva gargles. But I'm very excited this study, starting with Tavio, Andreas, and Alfonso immediately, in Brazil, where we're using the Bruker Biotyper and we're analysing matched swab samples where they have sequenced the samples which are positive, which had high CT values, and we're going to look at how we can pick up the P1 variant if that's very different, and also how well this can impact on saving lives essentially in Brazil. So I'm coming back to why. Why do we want to do this? Why is MALDI-Tof going to prove to be important, or I believe it's going to be proved to be important in what we're going to be facing in pandemics and future biosecurity. The whole point is we want to do population screening of the people without symptoms. This is to identify the asymptomatics who may spread the virus to others unknowingly, completely unwittingly, because that's how viruses are spreading. Now, you can look for viral antigens via the lateral flow department, but the sensitivity is probably too low. It's above the transmission threshold. RT-PCR is fantastic because it's way below the transmission threshold, and you're looking for the genetic information, the DNA signature of that virus. What we're proposing is to look for the antigen and an immunological response, i.e. the IgA elevation that's occurring within the sample. And we think that this is where MALDI-Tof will sit in terms of being able to detect symptomless, asymptomatic spread of pandemic viruses. Thank you very much indeed.
So thank you very much for the presentation. It was really interesting and kind of really current problem with COVID, definitely. We have one question from the chat from Anna. So very interesting presentation, thanks you. Do you have experience performing MALDI in nasal pharynx samples directly from transport media?
No, we want to do this. Initially, we were very hell-bent on just doing gargle, and we realized, well, we're stupid. You know, we need to be looking at the same samples. We had assumed that the transport medium would destroy the protein, but actually in some of the experiments we've done, very preliminary, it doesn't. We still see the proteins there. So we very much want to do that, and we very much welcome collaboration with groups across Europe to do this.
Okay, so I have on my side, I have a few questions for you. I'm not a virologist, of course, so but maybe my question will be a bit stupid for the presentation. I just noticed that you were looking for really heavy proteins. Like, you know, but normally we're looking at the 220 kilodalton range in bacteriology. What did you investigate already this part of the spectrum, or you not even try to look at it just to find some stuff in it?
We did look. There are markers in there. And I think there's still mileage to look in the smaller region. However, we really wanted to target this against the spike, and we really knew that we were dealing with the spike up in the higher mass range. It was sort of conceptually more understandable to a lot of people. We have, although I feel like I'm with my people here, they understand markers that, you know, it's a peak that is characteristic. Trying to sell that to the rest of the world is very difficult to people who are not involved in MALDI and bacteriology. They just don't get it.
Yeah, I can understand. In the meantime, we get another question from Thomas. How do you estimate the sensitivity limit of detection for patient sample? Okay, it was a really long question. In fact, there's different question inside one question. I mean, you can read it at the same time, and I so.
The clinical sensitivity, that is a real issue. That's why we are the study that we've done in Northern Illinois is really key to that, because we're comparing RT-PCR results with our results on the same on the same patient with samples taken at the same time. So we know we're equivalent to RT-PCR when we're looking for the IgA response and the S1 marker. The other compliance type studies we did for sensitivity, they were in vitro and it's not real. And I totally agree with them. It's a real problem. You know, you can do a lot of compliance work on in vitro mock samples, but when you get into the real world, it's different. You get a different pattern, and you really want to do the more difficult thing, which is look in the real world. Hopefully that's answered Marcus's question.
Okay, and so the second part of the question, if only the immunological response or the general multispectral variation will be analyzed, how would it be differentiated from the other infection?
Yeah, we're not just looking for the immunological response. It's the immunological response, i.e. in elevated levels of IgA heavy chain, and in combination, the spike coronavirus peaks have to be seen for it to score positive. Because what we've noticed is that we can find other virus peaks. And in fact, we did a wonderful study where somebody came in and gave the entire lab a cold. We saw rhinovirus peaks coming up, and you get an IgA response as well, but it wasn't coronavirus. So that's why we have to have the coronavirus spike proteins and the IgA response, the IgA elevated peak.
Okay, so we also get last one question from Mikhail, really active person in the chat apparently. Great work, Ray. Did you think a better resolution could be achieved in negative mass mode?
Ooh, at the moment, no, but I haven't tried it. And you know, everything's in positive mode. A little bit of a secret here: the buffers we use to extract from the lipids, because I think if we're in negative mode, we'll get a lot of lipids coming down and large complexes. So my guess, though I haven't proved this, is that we're better off in positive mode at the moment, because our buffers get everything to fly in positive mode. We are using a detergent in there that is mass spec friendly. You can't do this with STS, you can't do this with Triton, but these particular ones are very good.
Okay, well, there's no more questions, so I think we should unfortunately close. Oh, well, sorry, there's one more question. There's a remark from Maureen. Okay, it's more remark. Ray, I've done some work on the detection of COVID directly in the nasopharyngeal swabs using MALDI-Tof with sample from Kenya. She will be happy to discuss about it.
That would be great. We really, you know, value our collaborations across the world.
Okay, well, perfect. So, like I said, just a few seconds before, unfortunately, we have to